Morning, everyone, and welcome to Grand Rounds. Again, thank you for all spending an hour with us of your time. And I just want to say that I am very excited. This is the second of our chief resident Grand Rounds, and there, as I've said before, they're almost always some of our best Grand Rounds. So today, our Grand Rounds is by Dr. Chodara. She is going to be staying on with us, thankfully, in rheumatology, and her Grand Rounds is a gut feeling, the microbiome in inflammatory disease. Now, I just have to say, chief residents, they can solve any problem at any time of the day. They're on call for everything, and it could be something really mundane and something life-threatening and serious. So, in true style of the chief resident's stressful life, the whole audio-visual system in the VA went down today. So I just want to say that Dr. Chodara is going to be presenting Grand Rounds. She has her own laptop here. The system is going to work, but nothing stressful like doing a major presentation with no appropriate audio-visual things. And Clint, thank you for getting everything set up as you have been. So, Dr. Chodara, I just want to talk a little bit about her. Her mom is also in the audience here today. She did her undergraduate degree here at University of Wisconsin and had a double major in both genetics and Italian. She went on to do her medical degree here as well, which was followed by her internship and residency, and we were very honored that she was able to stay on and be a chief resident with us here. She has had many honors in a very brief time of her early career, and I want to highlight that she is an Alpha Omega Honor Society member, which is absolutely fantastic. She's had two publications and posters in her residency, which is quite amazing, and has been the first author on a paper, Managing Cardiovascular Disease and Rheumatoid Arthritis, Clinical Updates and Three Strategic Approaches, which was in the current rheumatological report. I think that bodes well for us here at University of Wisconsin. She's also been involved with service activities here and has been a member of the Internal Medicine Residency Wellness Committee. I know that we're hoping that after a fellowship here in rheumatology next year that she might decide to make University of Wisconsin her forever home for her career. And with that, welcome.
We have met, and we are not alone. And what I'm talking about is the title of my talk, the gut microbiome. What the microbiome is, it's the ecological community of the commensal, symbiotic, and pathogenic organisms that share our body space. So as Dr. Troberg mentioned, I studied genetics in undergrad, and I've always loved microbiology. And when I was researching this topic, I am still amazed by the sheer scale of what goes on inside our cells, how it might relate to disease, which I'll tell you about today, and just how much we really don't know. So I hope that you'll take away some of the sense of wonder that I had in exploring this topic and just be aware of kind of what it is, because it's going to be something that affects all of our fields in the next few years. I can pretty much guarantee that. And I hope you have a couple tangible takeaways. So we'll start with a question. So I'd like a show of hands. How much do you think the bacteria in our gut weigh on average? Who thinks it's 1/10 of a kilo? Anyone? How about 1/2 of a kilo? That's a pound. Okay. How about 1 kilogram? It's like 2 pounds. Okay, and then how about 2? The answer is 2. That weighs 4.4 pounds of bacteria in our gut. That's a lot if you think about it. Now the microbiome is a lot of different big numbers, because what we're really talking about is populations. We're not just talking about individual cultures of single bacteria. It's thought that 95% of the microbiome is located in the GI tract. We think that each individual has a unique signature in terms of the gut microbiota. And we talk about absolute numbers. It's estimated that there's 10 to the 14th organisms that live inside our cells. And I thought this statistic was really neat, that it's thought that perhaps ultimately 90% of the disease that we have can actually be linked back to the gut in some way, shape, or form. So a lot of this is, relatively newer studies, right? Because we've got all this like metagenomic data. Well, it's actually been studied since the 1600s. This is Van Leeuwenhoek, who invented the microscope. That's actually his microscope. And this is a little bit of a closer image as to what that looks like. He was actually looking at his own oral and fecal samples under the microscope and noticing that they were different. Back then he was calling them animalcules. Those were the drawings of his bacteria there, but regardless, this is something that's been fascinating people for centuries. So I have a few goals for you today. I'd like you to be able to name 3 roles of the gut microbiome and define what dysbiosis is. We'll talk about potential mechanisms of dysbiosis and a few conditions. I'm skewed towards rheumatology, but we'll talk about others as well. Specifically, we'll talk about spondyloarthritis, rheumatoid arthritis, and lupus. And then I want to give you some clinical applications of this fairly basic science field with the goal of improving patient care. Like I said, microbiome's so hot right now. So to do this, we'll talk about the gut microbiome. We'll spend a portion of time talking there. We'll also then move specifically into the examples I mentioned in different diseases. And then we'll talk about some lifestyle factors, some specific interventions, and then the idea of drug response and pharmacokinetics, because I think that's really compelling. So let's start with the gut microbiome. Now, it's thought that when we're born, our gut is sterile, but by age 2 to 3 years, that's when we've got all of our communities. And it's established pretty quickly, just by how you're delivered and by how you're fed. We know it's linked to your adult composition. Breastfeeding itself has been linked with less incidence of asthma, celiac, and ankylosing spondylitis, and other autoimmune diseases. And we've also seen that disruptions at a young age, for example, antibiotics less than 1 year of age, they actually increase your risk of childhood inflammatory bowel disease later on. So we do see that disruptions early on have lasting effects. We know there are genetic components there. There are some heritable taxa. In twin studies, we've seen that monozygotic twins are more similar than dizygotic twins, who then are more similar than if you look at mother and offspring pairs. And then there are a lot of influences, which I will get into, but we talk about environment, antibiotics, medications, and a lot of our lifestyle choices that we make. So the gut microbiome, once it's established by a couple years in, it's thought to be generally stable in terms of the populations we're thinking of. There's an interesting idea that perhaps the resilience of the microbiome, when there is an insult, whether it's antibiotics, a GI illness, or something else, the ability to take that hit and recompose itself, that might be a better indicator of health than just the exact makeup that we're talking about. We've seen some gender differences. A lot of this is in mice studies at the moment, but there's really thought to be potentially a hormonal role in terms of what bacteria might be expressed. And we also know that women have enhanced reactivity in terms of the immune system that has the benefit of women having lower risk of infection, but of course we know that women are at anywhere from 2 to 10 times more risk of autoimmune disease in general. And another way to think of this is as a collection of ecosystems. So the oral microbiome is different from the stomach, which is a pretty inhospitable environment. That's different from the duodenum, which is different from the colon. So it varies along the GI tract, but also cross-sectionally. I'll talk about the anatomy of the gut barrier in a minute, but you'll find different bacteria at kind of different levels too. So this is an example of a healthy microbiome, and this is what a lot of the studies look like. So I want to point out a couple of the big phyla we're looking at. So again, we're talking pretty broad categories here. We're not down to the species level at all. So firmicutes is a group of gram-positive organisms. Bacteroidetes are gram-negative organisms. And you'll see that together they make up about 2/3 of the whole microbiome. Again, this is a healthy version. I'll talk about dysbiosis in a lot of conditions, and oftentimes what we'll see is the proportions of those two phyla get flipped. So we look at relative abundance because we're looking at individual populations. So that's an important point. And then just to be familiar with these studies, a lot of times they'll talk about operational taxonomic units. And what that is a researcher will set a threshold, say 97%, in which they can actually group similar groups of bacteria. So that's the way they're usually referred to. Now, I told you the gut is pretty similar once it's established over the lifespan. I like this image because it shows a little bit of the changes that we have as well in early and end of life. So the red and the white are the two big phyla we talked about. So you can see that in an 8-week-old infant whose complete microbiome isn't yet established, it looks different than a young healthy adult who's got that kind of ideal proportion there, who's different from someone who's a frail elderly person who also start to see a little bit of changes there as well. And there's a lot of things that go into that: metabolism, medications, and other things. So then we talk about what exactly are these bacteria doing for us. Now, there's a few big roles I want to mention. Number one is nutrition. We've seen that these bacteria are able to synthesize vitamins. Vitamin K, a lot of the water-soluble vitamins, vitamin C, biotin. And in part of their commensal role, they provide these nutrients to the gut epithelium in which they reside. There's a lot to do with metabolism as well. Those two phyla I mentioned, we know that they have a lot to do with breaking down fats and processing complex carbs. And one of the most popular breakdown products to look at are called short-chain fatty acids. These are things like butyrate, propionate, but they're thought to actually have an anti-inflammatory role, so that's something looked at. And that whole area of study is called metabolomics. That's a whole nother area we won't talk a ton about today. But these short-chain fatty acids have a role in energy metabolism, even glucose and lipid homeostasis. They're formed through fiber fermentation, and they have a role in influencing satiety, so there's some role in body weight there. And we've also seen that reduced levels of these short-chain fatty acids are actually linked to bowel inflammation. And then most importantly, I would argue, there's this role in the immune system. So, of course, we're trying to balance self-tolerance with defense against invaders. And if you think about it, the most antigenic or foreign stimulation we're getting every day is by what we eat. So by physical presence and functional presence, the normal bacteria are able to outcompete pathogens. Of course, if you have reduced diversity or get knocked down by antibiotics, that allows pathogens, for example, C. diff, to bloom and cause pathology there. There's influences on innate and adaptive immunity. And of course, we know that gut-associated lymphoid tissue makes up about 70% of our immune system. That includes Peyer's patches, lymph nodes. So I told you that 95% of our bacteria are in the gut. Well, most of the immune system's there as well. And if you start to look into this, you will find so many more proposed roles. There have been evidence that these bacteria are able to produce neurotransmitters. So there's potential that it may be involved in mental health, in human behavior, and I also briefly mentioned weight. So like I said, if you keep looking, you'll find a lot more ideas. So this is the most immunology we'll talk about today, but I wanted to establish what the barrier of the gut actually looks like. So this is a layer of epithelial cells here, and then there's a mucus layer that is established by the normal good bacteria. These are two examples of potential pathogens. We've got Clostridia, we've got segmented filamentous bacterium, in some studies that's pathogenic. So they produce these microbial-associated molecular patterns. These are things like lipopolysaccharide, peptidoglycans, which are recognized by toll-like receptors located outside of those epithelial cells, but also on the antigen-presenting cells, I think a lot of us are familiar with, and subsequently lead to immune system stimulation. A lot of the studies will talk about kind of the balance between T regulatory and Th17 cells as influencing autoimmunity. And I liked this image because it also included the beneficial short-chain fatty acids there. So just to remember that we've got kind of the balance of the good and the bad there. So then we go into speaking about how do we actually study the microbiome. I told you it's a relatively new field in terms of the advanced technology we have, and it's really been in the last 13, 14 years that we've been able to develop metagenomics and actually study these things. So the US had its project aiming to describe the microbiome start in 2007. Europe had its sister project in 2008. And the two big techniques I want to talk about are 16S ribosomal RNA and metagenomic shotgun sequencing, because that's what mostly all of these studies use. 16S rRNA, that's produced from the 30S subunit, so it's actually specific to bacteria. So these studies are able to describe huge populations, although they don't get terribly they're not able to get down into the genus or species, so a little bit limited resolution there. Conversely, metagenomic shotgun sequencing, more expensive, not surprisingly, but it's able to get down into the family genus level, so we get a little more taxonomic resolution, and it has the benefit of describing all the other domains that I'm not talking about today. So we're talking about bacteria, but there are phages, there are viruses, there are archaea that we're still working on describing, and we don't know exactly what they're doing yet. So it's thought that by now we actually have our parts list, or kind of the description of what a normal microbiome looks like, mostly complete for the prokaryotic domains, so the bacteria, but not those other ones. So that's kind of the next step. So why is this all important? Well, immunosuppressives are expensive, specifically speaking to rheumatology. They have toxic side effects, and I'm not talking about that kind of steroids. We've all seen the effects of them. But we really need to identify effective therapies, looking for biomarkers to detect disease early, to detect disease progression, as well as exploring other methods of disease management. And really, patients are asking. I've been in clinic, not just rheumatology clinic, but when patients come in with chronic disease, they're looking for any way they can help self-manage their disease too. And it's empowering to be able to speak to a certain diet or something else that might be able to help them feel better and also help their disease. So that's what this ultimately is getting down to in my mind. So let's talk about dysbiosis and inflammatory disease. I'll give you a couple examples here. Now, what it actually is a departure from healthy. It's characterized by reduced diversity. We get pathobiont expansion, so expansion of those pathogenic organisms. Subsequently, we lose the commensal phyla. This is often taking place in a genetically susceptible host. We know a lot of the risk alleles for various diseases, and these tend to be the studies we're looking at. So the idea is that you get overgrowth of a certain bacteria that ultimately leads to inflammation via immune system stimulation. So I'll talk about the mechanisms here. But we've seen it in a lot of different diseases. Again, if you start digging into this, you will find a ton of information. So the three I'll be speaking about today, spondyloarthritis, lupus, and RA, but we've seen it in diabetes, obesity, fatty liver disease, even cancer, and smoking, and more if you continue to look. So in rheumatology, part of what we look at in trying to detect preclinical disease is when does that break in self-tolerance occur? Does it take multiple hits? What's going on there? So there's this idea that maybe the beneficial bacteria in our gut might trigger autoimmunity in someone who's genetically predisposed. So we talk about a few different mechanisms to know. Now, molecular mimicry is one idea where bacterial peptides actually just look like a self-peptide, and they lead to kind of fooling the immune system and causing stimulation in that regard. Citrullination, I'll talk a little bit more about later. All it is a one amino acid substitution from arginine to citrulline, but that is detected as foreign and causes a whole lot of pathogenic effects, particularly in rheumatoid arthritis. Some of these bacteria produce super antigens, so they just go on to stimulate the immune system more than normal. There's also this idea of epitope spreading. So normally we have an MHC class 2 molecule detecting one particular protein. Well, something happens that molecule is able to detect a ton of proteins, which just causes more downstream stimulation. Get autoactivation of these self-reactive T and B lymphocytes and amplification, production of inflammatory cytokines, autoantibodies, and disease in that regard. And interestingly, we do see 10 years of preclinical disease in a lot of these conditions where you'll see these autoantibodies, but people might not have symptoms. So there's some sort of underlying pathology going on, and that's what this is trying to get at. Another fact is that when we compare the patterns of dysbiosis, so I showed you the normal just a little while ago, when we look at the differences, they actually are pretty similar between a lot of these inflammatory diseases. So comparing the types of inflammatory bowel disease, multiple sclerosis, RA, they have pretty similar patterns of dysbiosis. So I mentioned in terms of diversity, that's one potential indicator of health. Well, this was a study looking at about 68 patients who they did 16S rRNA sequencing, one of those techniques I mentioned. They looked at a population of control, healthy people, those with spondyloarthritis and rheumatoid arthritis. Here they just looked at numbers of species, and you can see that the two inflammatory conditions were not significantly different from each other. However, compared to normal healthy people, there were a lot less species. So we see lower diversity there. And then there's the idea that when we get towards proof of concept, a lot of this is done in mice to start out. But what we see in a lot of these conditions is that germ-free mice actually aren't getting the diseases. So a couple examples here. This is looking like RA, autoimmune arthritis. We have lupus, diabetes. This is supposed to be ankylosing spondylitis in a mouse model. When they're raised in a germ-free environment, so none, not even the good bacteria, the impact on their disease, a lot of them don't end up getting disease. Lupus is interesting because there's no change, but there seems to be some sort of correlation there. So I'm going to first start by talking about spondyloarthritis. And just to refresh, that's a group of multiple conditions, oftentimes affecting the sacroiliac joints. We've got ankylosing spondylitis there, psoriatic arthritis, and it's often characterized by disease in multiple mucosal sites, including the eyes. So a systemic disease with arthritis is a prominent part. So to speak a little bit about that, we know that there is a big link between inflammatory bowel disease and spondyloarthritis. Up to a third of patients with IBD will have some sort of arthritis, and it goes back the other way. We've seen reduced diversity in a number of these studies. So the microbiome is just, there are fewer species around, potentially allowing pathogens to take over. Again, those germ-free mice who are genetically susceptible, they have that HLA allele. If they're grown in a germ-free environment, they don't get arthritis. But if you go and introduce what we think are normal, lactobacillus, for example, to those mice, they end up with the usual progression of their disease. So they get the arthritis, they get the sacroiliitis. So there's something to be said for introducing that bacteria and what effect it might have. I would actually group reactive arthritis in this category as well, for the reason that, we're talking about perhaps the idea of gut permeability here as well. So reactive arthritis is the syndrome that can occur after a GI illness, for example, salmonella or shigella. You get inflammation there, and somehow there is a cross of the barrier, because we've actually been able to detect DNA from these infections in the joints themselves, and leading to a systemic syndrome. This is just an example of the many sites that can be affected in reactive arthritis. So there's some way for it to get across the gut barrier and actually cause disease in multiple areas. The other entity to bring up is gastric bypass-associated arthritis. This is a rare entity, but what I think it actually shows is that, this is an example of the Roux-en-Y anatomy here. So you've got this whole area of bypass small intestine, and I'm telling you about these ecosystems, these populations there. There's a thought that you get some sort of dysbiosis that occurs here, perhaps because it's not being stimulated, but regardless, what ends up happening is you get immune complex formation and deposition in joints. Again, something to be said for the gut barrier being compromised in that regard. So the joint and gut are linked. A nice study in the 90s actually looked at ileal biopsies from patients who had spondyloarthritis, and they found that half of them actually had microscopic gut inflammation, but they didn't have any symptoms. And they found that when they tracked them and rebiopsied them after treatment, as the joint inflammation improved, so did their gut. This was a nice study as well. This was from 2014 by a Belgian group. They looked at 68 patients, and they biopsied them. They all had spondyloarthritis, and they had a group of healthy patients as well. And what they found was people who had chronic gut inflammation, that's that category there, they had significantly increased, this is bone marrow edema, of their sacroiliac joints seen on MRI. And the y-axis here is the SPARC score, which is a radiologic image of damage seen on MRI. So those who had chronic inflammation had worse joint inflammation on imaging. And I think that just supports this idea of treat to target, which is pretty broad across rheumatology, but the idea being that you really want to completely abrogate any inflammation that you see. Because in all likelihood, we are having a lot more effects on the gut, perhaps, than even just on the joints that we're following. A couple more specific examples, and these are two populations that were identified as being increased in these spondyloarthritis patients. Dialister is one. It's a member of that Firmicutes phyla. So it was found in looking at patients who, again, had spondyloarthritis and they had ileal biopsies. Those who had chronic gut inflammation, it actually correlated as you had more of this dialister, their disease activity score was higher. So they found it was more abundant in disease, and as your disease worsened, you had a higher proportion of this bacterium. Similarly, Rheumatococcus novis in a different group, this is Brevon's group, they found, again, it was increased in proportion in people who had spondyloarthritis and IBD particularly, so kind of that double hit there. But it also, again, this is a disease activity index, amount of rheumacoccus. They found it increased as well. So they proposed that perhaps this might be a sort of microbial disease activity marker. It's not quite there yet, of course. This is a lot of basic science, but there's something to be said for these increased populations that we don't see in healthy people. So next I want to talk about rheumatoid arthritis, that being the systemic condition causing a lot of peripheral joint arthritis, erosive disease, but can also have multiple systemic effects as well. So even back in the 70s, we were looking at the potential of bacteria being involved. A Swedish study actually they were able to isolate this Diplostreptococcus. Streptococcus. They found it in 80% of people who had RA, only 20% of healthy. It's also found in pasteurized milk. So what they did was they actually injected it into rats who subsequently developed arthritis, started making rheumatoid factors, so they said it caused RA. Now that's not exactly true, but there's been this idea that there's some sort of link for many years. We see reduced diversity in early RA. This will be a theme you'll hear. And then now I want to talk about citrullination, and with a shout out to Dr. Shalef, whose lab focuses on this, and I just wanted to say a couple words about it. Now I mentioned it's a 1 amino acid substitution, but we know it's a critical step in rheumatoid arthritis, and it often occurs years before we see any sort of symptoms in people. The CCP antibody, the citrullinated antibody, it actually correlates with disease duration as well as damage. And we've seen evidence that citrullinated proteins are created in the lungs, especially in people who smoke, and are also related in periodontal disease. So we're going to go a little proximally and talk about the oral microbiome for a minute here, because RA does have a strong link with periodontal disease as well. This was a paper in 1900 in the British Medical Journal, actually proposing that dental disease was the root of all disease, including arthritis and multiple other disease. Now, Hunter wasn't completely wrong, I'll say that, because Porphyromonas gingivalis is one example of a bacteria that is increased in dental disease. We know it's there. And it actually produces the enzyme peptidyl arginine deaminase that citrullonates. So here we have a bacteria that can actually create these citrullonated proteins that we know to be an effect in rheumatoid arthritis. And 119 years later, also in British Medical Journal, there's a nice study that just came out looking at P. gingivalis that actually was able to show that it induced rheumatoid arthritis and produced CCP antibodies in the rat. So we're starting to see some proof of concept there, which I think is really fascinating. And I like this image with this idea of multiple microbiome hits potentially breaking that self-tolerance and triggering something like RA. Here we've got the P. gingivalis. I told you it can citrullinate, and then you get this kind of inflammatory cascade leading to RA. We're going to talk about the gut microbiome again, and specifically this Prevotella copri here too. So that's another one that has been proposed to be involved. So this is one also in that Firmicutes class, and what I want you to pay attention here, this was a nice study by Shear done in 2013. He looked at about 120 people. They did 16S ribosomal RNA sequencing on them, and they identified that this Prevotella species, that's the red one, was increased in people who had new onset rheumatoid arthritis, so people who had not yet been treated, and it was looking a lot different than those with healthy. It's also actually been described in a couple other diseases as well as being increased. So here, simply, they're looking at the number of total reads and a few different conditions. So we've got the new onset RA. You can see a lot of the red there. There's healthy subjects, a lot less. Chronic rheumatoid arthritis, and they also looked at psoriatic arthritis. So they saw a difference, proposed that there. The other point I wanted to make, though, was that it's very interesting that the chronic RA folks who had mostly been treated in this study and had their disease controlled, they're pretty similar to the healthy people. So then there started to be this idea that maybe our treatment is actually helping to normalize the microbiome. There are other bacteria that have been linked to RA. I'll just briefly mention them. There's a few here. We've got Collinsella that actually induced arthritis, caused inflammation in a rat, Proteus and UTIs, Epstein-Barr virus, mycoplasma, and EBV itself has actually been shown to citrullinate, so potentially causing pathology there. But in all likelihood, it's a molecular mimicry type mechanism for a lot of these. And when we actually look at the joint fluid of people with rheumatoid arthritis, we have been able to see different cell wall constituents, things like lipopolysaccharide, peptidoglycans, as well as bacterial DNA. So I mentioned this idea of the microbiome potentially normalizing with treatment. So this was a nice study. They actually looked at, this is people with inflammatory bowel disease, and they looked at who responded, who partially responded, and who didn't respond to a TNF inhibitor. And this was at time 0. So these are the non-responders here, and this is just looking at the diversity. And so here they mentioned that at time 0, the non-responders had much lower diversity. And then when we get to time 3, when they all had been treated with a TNF, that difference was no longer there. So they proposed that perhaps that was representing improvement of the microbiome, or at least a loss of that difference there. So in another study, this was a really nice one in Nature Medicine from 2013. They looked at people with rheumatoid arthritis who were on methotrexate and etanercept. They were also able to show that the microbiome partially normalized. And what they ended up doing was then they made these training sets. This is from a fecal microbiome set, but they actually tried to go backwards and say, by analyzing the microbiome itself, are we able to say which of these patients actually had rheumatoid arthritis? And this is the receiver operating curve, and you can see that it's actually got a pretty decent sensitivity and specificity for trying to predict who has disease based on just looking at the microbiome. So some pretty interesting ideas there. So then we'll move into talking about lupus. Definitely the most complicated of the bunch, I would say, and with a lot of systemic disease, particularly renal disease is something we talk about, and a lot of autoantibody production. Another interesting historical fact, in the 1930s, anti-DNA antibodies were first reported in patients who had bacterial infections, things like strep. Only in the 50s did we learn that they're actually related in lupus and actually can cause significant pathology, like the renal disease I mentioned. We see decreased diversity, again, just contracted ability of the flora to react there. And there's a big link between infections. There's a whole list of infections that we know to trigger lupus, whether it's triggering onset of disease or causing flare. But there have also been reports from patients that having disease actually leads to improvement of their lupus symptoms. And maybe that's because of redirection of the host immune response. We've also actually seen that some infections are even protective. So there was a study looking at African American patients, and those who were H. pylori seronegative, that was actually associated with earlier onset of lupus and increased incidence of lupus. And areas of the world where helminths are endemic tend to be developing countries. There is increased incidence of autoimmune diseases, not just lupus. In all likelihood, that's related to the hygiene hypothesis. We've also seen that antibiotics can trigger lupus flares. We know a link between sulfa antibiotics, and whether that's an immunomodulatory mechanism or potentially affecting the microbiome, we aren't able to say yet, but these ideas persist. So this was a nice image from a review that was comparing the normal healthy gut. So you have kind of your normal flora, shapes the B-cell repertoire, the different antibodies, but in lupus, having this restricted diversity allows for potential pathogens to bloom or take over. So the two I'll be speaking about might sound familiar, Rheumatococcus, I mentioned that back in Spondyloarthritis, and also Enterococcus gallinarum. So there's a couple different mechanisms they talk about, but the idea is that they've been able to show some translocation and ultimately immune system stimulation there. So first we will talk about E. gallinarum. This was in Science last year. They orally gave this bacterium to lupus susceptible mice, and then they were able to actually sequence the bacteria from different spots in the body, mesenteric veins, lymph nodes, liver. Somehow it got across. And in these mice, it ultimately induced double-stranded DNA antibodies, so some sort of lupus pathology there. Then they did a couple more studies where they either gave them oral vancomycin or they vaccinated them with a heat-killed strain of E. gallinarum, and that actually reversed the autoantibodies and improved their mortality. So there was some sort of benefit by trying to treat this specific bacteria. So then they went to look at humans, and the way they did that was they took lupus patients who also had autoimmune hepatitis, so some liver involvement, and they looked at their liver biopsy results. They compared those to healthy cadavers, so a little bit of a different population there. But they were able to show that you could only see this Enterococcus DNA in the people with in the lupus patients who had the liver biopsies and not in those healthy cadavers. So image from a PCR, this is the E. gallinarum. You can see the band there. We've got the lupus patients here, and it's only present in lupus patients. And this is comparing the proportion of that E. gallinarum in the liver biopsies. So we've got the control group, autoimmune hepatitis, significantly increased, and then this is non-autoimmune cirrhosis there. I've spared you from some of the big, broad bacterial pictures, but I just wanted to bring up this one, looking at relative abundance here. So the red is Enterococcus. This is an example of the controls, so much less red when you look at it compared to autoimmune hepatitis. That's how they're interpreting these. So in a somewhat similar fashion, just earlier this year, a group out of NYU looked at Rheumatococcus novis, which I already mentioned in spondyloarthritis, but they found that it actually was increased in abundance in lupus patients. They looked at a decently sized cohort of female lupus patients. It was about 61 patients. They had varying levels of lupus activity, and they found like 5 times more incidence of Rheumatococcus compared to healthy controls. And they found that it correlated with double-stranded DNA, so there was actually a direct relationship there. They did some mechanistic work and proposed that it might be part of the cell wall that actually was a toll-like receptor agonist, so potential mechanism. And they also looked at levels of fecal calprotectin in these patients. Now, fecal calprotectin we often think of in inflammatory bowel disease. It's an accepted indicator of an impaired gut barrier, but in a lot of these lupus patients, they found elevated levels there, so something was going on. This is looking at, again, operational taxonomic units. I mentioned those kind of similar groups. What this is trying to show is decreased diversity, again, in the lupus patients compared to healthy controls. When they divided lupus into activity, significantly more people who had worse lupus had lower diversity there. And then they looked at the anti-Rheumatococcus antibodies here. We've got the IgG. And they looked at lupus patients, divided them into who had renal disease, who didn't. And those who had renal disease had a significantly higher proportion there, too. So again, we're thinking about this idea of potentially bacterial markers, which I think is really fascinating. And overall, what I want to say about that is that we know there's a balance. This is kind of the ideal balance. We've got normal, healthy gut. We've got a good balance of the pro and anti-inflammatory cytokines. And when we have dysbiosis, which we have observed, that something shifts to support an autoimmune state. So we get some sort of overgrowth, but the pro-inflammatory cytokines, those are really shifted up. So the question becomes, is this cause or effect? Is it the gut bacteria themselves that are driving autoimmunity? I think that we're starting to see some evidence that they very well could be. But the other question is it just a general inflammatory state that actually in itself is shaping the gut microbiome? Only time will really tell in terms of what exactly we'll see. So then let's move on into some of the practical considerations. We'll talk about a few different ideas here. What we're really talking about at its base is modulating the microbiome. So we've really been doing this all along, even if we don't know it. Traditional herbal medication, even centuries ago, actually has an effect, and now we're doing things like fecal transplant. So we can think of them as targeted approaches. Probiotics is one example, antibiotics as well, where we're giving one specific strain and having an effect there, or untargeted methods. That's where fecal transplant comes in. You're trying to shift someone's whole microbiome into someone else. Diet, in all likelihood, as well, and medications, as I'll show you. So diet, when we think about prebiotics, this term is big. All it is food ingredients that can stimulate growth or activity of gut bacteria. This is like all the micronutrients that we have. And these are foods that are rich in prebiotics. Probably not surprising that it's a lot of fruits and vegetables, the things we're supposed to eat, right? But this was a nice study actually looking at the contribution of fiber, and I think fiber is a really big part of this because I mentioned earlier that some of the breakdown products of fiber, namely those short-chain fatty acids, are in all likelihood anti-inflammatory and have an effect there. This study was looking at mice, and they were able to show that if you fed these mice a fiber-rich diet, they had a normal barrier, all was well. If you gave these same mice a fiber-free diet, you just did not have a good barrier. Those normal microbiota were not able to produce the mucus, keep that barrier intact like they were supposed to. Or if you grew them in a germ-free environment, so they really had no bacteria, whether you gave them fiber or not, you just didn't have evidence of a good barrier. So this is really the idea that it's an important part of what our diet should be. Thinking a little more about specific evidence there, specifically in rheumatoid arthritis, there's been a lot of small studies. Oftentimes they'll start with like a 7 to 10 day subtotal fast, and then they'll do their diet intervention. There's an idea that maybe even the subtotal fast actually induces a transient immunosuppression, and the mechanism for that might just be that you're getting less antigenic stimulus, less foreign protein, and so that actually calms things down for a bit. But regardless, we've seen clinical and functional improvements, some weight loss, some lipid improvement, but when they do look at the microbiome, they don't see specific changes in flora. One study looked at the nurse's health study, and they found that a healthier diet was associated with reduced risk of RA, with a pretty compelling hazard ratio there. In the other conditions I've talked about, in lupus, it's more been looking at specific micronutrients, things like vitamin D or E, omega-3s. Maybe those decrease some disease activity measures, maybe they alter the flora, but it's not great data. And again, in spondyloarthritis, one study looking at high-dose omega-3 fatty acids found an improved disease activity index, but others have found really no effect. So we definitely don't have enough information to say. But people will come to you and ask, they will ask me, what is an anti-inflammatory diet? What can I tell them? My answer would be it's probably Mediterranean type, and I'll tell you why. There was a randomized controlled trial back in 2003 looking at RA. They put people on the Mediterranean diet for 12 weeks, and they actually found improved disease activity measures and no change in the control. When they looked at their fatty acid profile, they were able to see that that improved. So in all likelihood, we're increasing those levels of short-chain fatty acids there. Just to remind ourselves what the Mediterranean diet looks like, the base is being active and sharing meals, but you've got fruits, you've got vegetables, the grains, olive oil, legumes. Then we get kind of the healthier meats, right? Got a little bit of wine in there too, but you know, the general Mediterranean diet. There was a nice study in Circulation earlier this year. It was a systematic review and meta-analysis that I took this actually from that study because I liked what they said. The available evidence is large, strong, and consistent that being able to conform with this diet is associated with better cardiovascular health outcomes for a lot of things. Coronary heart disease, stroke, cardiovascular disease. So these rheumatologic conditions I've talked about, you're already at way higher risk of cardiovascular disease just from the general inflammation. And being able to add a diet that perhaps reduces that, I think is really compelling. And then no talk about rheumatoid arthritis or lupus would be complete without mentioning smoking. It's a huge part of the environmental risk in rheumatoid arthritis, and we've seen evidence of those citrullinated proteins, as I've mentioned earlier. We see them in lungs of healthy smokers, we see them in early RA. If you have these susceptibility genes, you're probably just at even higher likelihood. We know smoking's role, of course, in cardiovascular disease, inflammatory bowel disease pathogenesis, particularly in Crohn's disease. And we've seen that the gut microbiome itself has been linked to elevated cardiovascular risk, possibly through an inflammation mechanism there. One study looked at diversity again, and they looked at the oral microbiome of smokers, and they found an increased incidence of this Prevotella group. So again, we've seen that in the gut, and now in smokers' mouths, we've also seen it. And they also looked at diversity. This is a Shannon index, which is an ecological measurement of diversity, and this is looking, actually, they looked at control people, they looked at people who were on e-cigarettes, as well as tobacco smokers. And the tobacco smokers had a significantly reduced diversity compared to everyone else. And then I mentioned dental care as well. So periodontal disease has a big role in rheumatoid arthritis, also in cardiovascular disease and other conditions. But dental care is hard to come by. Not all dental providers accept Medicaid or Badger Care. And I wanted to provide a couple resources just to be aware of, because this is such an important thing. There's a list of free clinics in Wisconsin through the DHS website, the Department of Health Services. And here at the VA, you're eligible based on your level of service connection. And also, if you're able to show that someone's dental health is affecting their health conditions, that may be an indicator as well to get some care. But if someone is really in need, there's this cool event called the Mission of Mercy that actually took place in Madison back in 2012 at the Alliant Energy Center. And it's basically like it's a big free dental fair that occurs over a day or over a weekend, and they try to serve as many people as they can. So it's not a substitute for regular good dental care, but it is something for people who might not have anything. And what it really comes down to, though, is for these high-risk populations, we really need improved coverage. So to say a bit more about oral microbiota, something going on at our own institution, we have something called the Cohesion Trial, which is actually in conjunction with the Marshfield Clinic. They're looking at this Plaque HD toothpaste. It actually is the one that turns your teeth this color. We've all probably seen it. And they're looking at high sensitivity CRP levels in adults who have mild or severe periodontal disease. And as a secondary objective, they're actually looking at the microbiome and what it looks like before and after this intervention. And I just wanted to mention it because we've got a few of our own involved in this. Karen Hansen is one of the PIs. Drs. Tattersall, Stryker, and Lemansky, also from UW, are also involved. So I look forward to seeing the results of that. And then, of course, with our own Wisconsin Institutes for Discovery and UW Biotechnology Center, we've got our own microbiome hub here. And they're looking at a whole bunch of different projects within this realm. It's a multi-specialty collaboration. Dr. Andes is involved in this as well, I saw on their website. But one of the cool projects they're doing is actually looking at precision editing of the microbiome using bacteriophages and how that might be in effect. So that's their website. I would encourage you to check it out if you're interested in that. But just another example of kind of what we're doing here at UW. So a couple more interventions I want to speak about. Well, probiotics are a big one that people really ask about. Unfortunately, one systematic review that looked at healthy people found no effects of different probiotics on microbiome composition. They also showed that they did not even engraft, so they literally just passed right through. In the conditions I've discussed today, we found pretty minimal or no evidence of any sort of benefit that we see. Now, I haven't talked much about scleroderma today, but it is a disease that has pretty significant GI effects and symptoms. There was a pilot study of just 10 patients, but they actually found some improvements in a few measures, including patient-reported measures like reflux or bloating and emotional scales. But what I think it really comes down to is we have our established gut microbiome, we have it there, and it's resistant to colonization by pathogens, yes, but also by these probiotics that might just be recognized as foreign because they're not similar. So I think a way that to perhaps get around this is in the future when we're able to better define what an individual microbiome looks like, perhaps then we'd be able to give tailored strains that are more similar to actually have the chance of engrafting within someone's gut and actually providing benefit there. And then of course we'll talk about fecal transplant. To remind ourselves what it is, it's literally taking stool from a healthy donor, putting it in a recipient, GI tract. Usually that's done via capsule form. It can also be done via enema. And there's an FDA exemption in recurrent C. diff for this treatment. And it's been shown that it actually increases microbiome diversity. So it has the intended effect. If you go to clinicaltrials.gov, you will find over 200 ongoing clinical trials in a lot of different conditions looking at fecal transplant. Anything from ankylosing spondylitis, which I've talked a little bit about, melanoma, irritable bowel syndrome even. So that's something that remains to be seen. Now, it's talked about a lot in inflammatory bowel disease, and I briefly wanted to mention, of course, the pathogenesis of IBD is a lot more complex than C. diff, which is an induced defect from antibiotics. A big systematic review that came out a couple of years ago looked at, in total, 122 patients, and they found overall a 45% remission rate after these experimental trials. It seems to be more effective in Crohn's disease compared to ulcerative colitis, but certainly we are nowhere near the point where this can be something we recommend. There is also a risk of flare that's been known with this treatment as well. So it is something that is out there, and I think we'll learn more about all these conditions as we go forward, but we can't say much about it there. And then there's this idea of super donors. So as we learn more about what our microbiome looks like, there are certainly people out there who've got the best microbiome, the richest, the most butyrate. And so it may turn out to be that not just anyone can donate their stool. So we will see. And then lastly, I think this is where it's going to come into affecting all of us in terms of drug response. So I'll first talk about some findings in rheumatology, and then I'll talk about a lot of other stuff. So there are a lot of antimicrobials that we already use in rheumatic disease: sulfasalazine, rifampicin, the antimalarials. And sulfasalazine, that's an image of it there, it was actually deliberately created in the 1940s to combine an antibiotic and an anti-inflammatory, back when we were still thinking that there was an infectious agent in rheumatoid arthritis. And even nowadays, we know triple therapy, which is 2 of these antimicrobials, is just as effective as methotrexate and a biologic in rheumatoid arthritis. A couple trials, actually a couple decades ago now, 2006 and 1995, one looked at adding doxycycline to methotrexate and found improved outcomes compared to just methotrexate. This was in humans, both of these. One looked at minocycline and found that that had some improved outcomes compared to placebo. Now, in all likelihood, it could be an immunomodulatory effect of the medication itself, but knowing what we know now about the microbiome, there might be some effects there. And then there's this idea of methotrexate. So not all patients with RA will respond to oral methotrexate, but some, when you inject it, then you'll get a response. So I've told you that there's dysbiosis in RA, right? So some of the populations that are increased are these Bacteroides species. And it's been shown that some of these Bacteroides actually have high levels of dihydrofolate reductase. So it might actually be competing with a host dihydrofolate reductase to actually cause more rapid metabolization of the methotrexate, which is perhaps why when it's given orally, it doesn't have as much of an effect. So when you bypass that by giving it injected, maybe it has an effect there. So I like this image because I think the microbiome is going to be something that we really need to think about when we're speaking about pharmacokinetics and drug effect. We've got the drug up here, we've got the host, the genetic factors, and we've also got the microbiome as its own entity. In metastatic melanoma, it's been shown that the microbiome affects who will and won't respond to these PD-1 inhibitors that are big now. Also in oncology, the fluoropyrimidines microbiome has been found to affect the response. Metformin is a significant contributor to dysbiosis in type 2 diabetes, and in all likelihood has a lot of effects there. In cardiology, there's a strain called Eggerichella lenta that actually metabolizes digoxin more frequently. It's found in normal gut microbiota, so this may perhaps contribute to why it's really hard to get normal digoxin levels in some people. Also in that realm, I mentioned a little bit that the microbiome has a role in lipid metabolism and homeostasis. In addition, when we give different statins, the microbiome actually affects which statins people might respond to. So overall, I would say that meds, because people are on a lot of meds generally, and we're not knowing all of the effects, they're probably the most important covariate in the composition of what's in our guts right now. There was an in vitro screen, this was in Nature last year. They looked at 1,200 human marketed drugs, FDA approved, and they found that half of the non-bacterial anti-infectives and a quarter of all of the human-targeted drugs inhibited at least one gut commensal. And this wasn't just things like proton pump inhibitors or H2 blockers. They looked at all classes, antihypertensives. Atypical antipsychotics were overrepresented. So what I'm saying by this is that when we prescribe medications, we're probably having even more effects than we can really know at this point. So then we get into kind of future directions. And we're still working on determining what exactly is a healthy microbiome. We also need to start looking at these other domains. I mentioned the viruses, the bacteriophages, the archaea, the eukaryotes, and how do these all interplay? We need to establish causality. A lot of these studies are observational. We're just simply at the point where we're describing what these populations look like. We don't actually know the role of a lot of these bacteria I've mentioned today. So that's a logical next step, as well as showing proof of concept. We're starting to see some of that in mice studies and more basic science there, but of course the next step is getting into humans. I think the idea of biomarkers is really compelling. There's one act right now looking at the microbiome in Crohn's disease and trying to predict, depending on what your microbiome looks like, if you stop a TNF inhibitor, who's more likely to relapse. So there's one example there. But also being able to detect preclinical disease as well as follow disease. We really need some ideas there. I mentioned this idea of the omics. So a lot of this we've talked about today is metagenomics, but we have metabolomics, which is looking at the byproducts. We have metatranscriptomics, looking at proteins. There's all these different fields. We have all this big data, and really being able to narrow it down and use that in precision medicine is really the next step. And I'd love to see bigger, better, more well-done studies looking at diet, looking at probiotics, even fecal transplant, and these other potential interventions that we can really do to try to help people. So in summary, I want to say we are really in the infancy of understanding this area of our existence. We really need to think of ourselves as hosts in the microbiome in its own right, which is definitely a bit of a perspective change. We have extensive influences. We know the microbiome has extensive influence on the immune system. I hope I've shown you that there's also roles in dysbiosis in terms of many diseases. And what I think a patient comes and asks, the things we can recommend are a good diet, probably Mediterranean type, keeping up with dental care if at all possible, definitely not smoking, and controlling inflammatory disease, because in all likelihood, beyond what we're able to see in the joint, what we're able to track clinically, we're probably having a lot more beneficial effects. And with a reference to one of the best albums of all time, I will say that as our technology advances, as we learn more, we're kind of opening Pandora's box in a sense, but I think that in itself is an amazing reason to keep pushing forward and trying to learn more about this area, because it will affect you in the future somehow. Thank you. I have to thank Christy Bartles and Bennett Oldham, my mentors, for all their help. Jeremy and Amy for helping me prepare this presentation. Nasia, Softar, and Karen Hansen for giving me their blessing on this whole microbiome thing. Mary Hitchcock with Eveling Library. My chiefs. I am lucky to get to work with some of my best friends. And of course, my fiance and my mom. Thanks.
That was fantastic.
Thank you.
So go ahead and call on the audience.
Dr. Vogelman.
Well, now you can go Pink Floyd Breathe and enjoy your evening. This was wonderful. There are some resource-rich nations whose populations are 10% at least on PPIs now.
Yeah.
Has anyone looked at this signal and what we're doing to our population? Because that's a real great acid suppressor.
Yeah, so Dr. Vogelman asked about so many of the population being on proton pump inhibitors, for example. There is research out there. It was out of the scope of kind of my talk today, but if you actually go and look into this, you will find that there are effects. And they found, thinking to some of what I've read about the GI-specific mechanisms, there are effects on the microbiome. In all likelihood, it's related to probably restricting some of that diversity. They've also seen it with NSAIDs as well, having an effect on the gut. And whether that's through an inflammatory mechanism or working on the microbiome, it's a little bit hard to say. But I think there certainly is research out there, and it's being looked at. Yeah, Dr. Mish?
Yeah, I think you said some mouse model with E. gallinarium causing arthritis.
Yeah.
Yeah.
So in that study, particularly in terms of the control, they used the lupus susceptible mice, but they just didn't give them an organism. So they were just looking at kind of proof of concept, actually giving this bacteria. Mm-hmm. Yeah. Dr. Perrins.
Well, I'm curious. So you said there obviously 80% of autoimmune disease occurs in women.
Yeah.
So is there a fluctuation in the microbiome across the menstrual cycle?
That I am not sure. That's a great question. So she asked, since there's a much higher incidence of autoimmunity in women, and we see some differences in microbiome between men and women, is there a fluctuation across the menstrual cycle? I would have to look into that. That's a wonderful question. Also, just with regard to being postmenopausal and changes there, I would think there might be, since there's an idea that hormones are playing a significant role, testosterone and estrogen, in terms of what populations we're seeing represented. So I would think there probably is a role. Mm-hmm. We just might not have defined it yet.
Thank you.
Thank you.
You did a fantastic